Hello again. Well, before we can get into metabolic biochemistry, which is where we're going with all this, we have to come to grips with the concept of enzymes, what they are, what they do, and kind of get an appreciation for the general uber importance when it comes to the ways that cells carry out their functions. And so I'm entitling this kind of like enzymes and catalysis. This is a good place for us to get a general start. The general term here is catalysis, which basically means that we're going to be speeding up some type of chemical reaction. Now, a key idea here is that with catalysis, what's going to be happening is that this chemical reaction normally proceeds at a certain pace. Okay? In other words, it's a naturally occurring reaction. It's a reaction that's favored by the universe, but it doesn't happen very fast, and we can make that happen a lot quicker with the presence of the catalyst. Okay, the catalyst is the thing that carries out catalysis. It's the thing that makes the reaction speed up. And I guess the key idea about catalysts is that they're not affected, they're not changed as a result of carrying out their catalysis. It's not like they get used up, right? A lot of times you might think, well, if we're going to be doing this thing, that must consume whatever it is that's doing the work. No, in the case of the catalyst, it's not being consumed. It's returning back to the exact original state that it was before it carried out the catalysis that it just completed, right? So you can call it a catalytic cycle. The catalyst is, at least theoretically, just as good at the end of its catalysis as it is at the beginning, and it's ready to start a new one, okay? So again, two ideas. One, catalysis is simply a speeding up of a reaction that would happen normally and spontaneously. And two, the catalyst itself is not changing as a result of having carried out the catalysis. So what we're really moving towards here, very quickly, I'm not going to talk about catalysis anymore, except for the fact that enzymes, things that we, these biological machines that carry out catalysis, these molecular machines that do this little seemingly magical trick of making reactions move forward, are simply doing a very specific form of catalysis, right? Enzymes, it comes literally from the word inside of yeast. And it kind of calls back to the discovery of enzymes, that there are these things, these entities inside of yeast cells that would that could be made to carry out these pretty substantial functions, right? And so the example that I want to give you here, and it's not an example from yeasts. It's an example from something we've been talking about in the past. We discussed carbohydrates and disaccharide structure, sucrose. Table sugar is a disaccharide. And we said that a disaccharide is made up of two monosaccharides. The two monosaccharides would be a glucose molecule and a fructose molecule. And if you put those together in with a glycosidic bond between those two things, in other words, you have a disaccharide, this would be sucrose. Okay. And we also know that the process of catabolism or hydrolysis of a disaccharide, in other words, if we take a water molecule, split the water molecule, and have the water molecule split into two, break apart the sucrose, we can end up with, as the products of this reaction, a glucose and a fructose as separate molecules, right? So glucose, fructose, two monosaccharides, sucrose, the disaccharide. This would be a simple example of hydrolysis. Okay. So hopefully this is a pretty straightforward and review for you. The other thing we remember about hydrolysis is that these are exergonic reactions, meaning that the end represents a lower energy state relative to the beginning, higher energy. Okay. And in order to go from the reactants to the products, we're going to actually have a release of energy. Because we have to account for all the energy that's there in the system. The main point here is that we don't have to put energy in. This is something that's going to be favored by the laws of the universe, by the laws of thermodynamics. And therefore, it's a little bit mysterious to us to say, well, why don't glucose, the two monosaccharides and sucrose, why don't glucose and fructose come apart from each other when you mix them with water, right? And you could all picture this. If you take like a teaspoon of sugar and mix it into a coffee cup with warm water in it, okay, you know that the sucrose is going to dissolve. But when it dissolves, it's not breaking apart. We're not undergoing hydrolysis then. It's just simply going into solution. It stays as a disaccharide throughout that cup of sugary water at this point, right? Why don't you get them breaking apart into the monosaccharides, given the fact that we know that this is something that's going to happen spontaneously? It's favored. It's not going to happen spontaneously in the sense that you're going to be seeing a whole lot of this happening, but this is something that's favored by thermodynamics. Okay? And so, in other words, you could say that the sucrose and the water wants to split apart to become glucose and fructose, reaching a lower energy state. That's what the universe is driving it towards, but it's not allowed to happen because, well, in this case, it's pretty easy, right? You know that in order for us to rearrange the covalent bonds, in order for us to actually run the this hydrolysis, right? We have to break a bond here. We have to break a bond within the water molecule, and breaking bonds requires energy to go in, and then when we come back to form the new bonds with glucose and fructose separately, we get more than that amount of energy back. In other words, we have to put energy in, okay, in order to break those bonds, and then we get energy back when we form the new bonds, and the energy back is greater than the energy that goes in at the beginning. And I guess the thing that you can realize here is that this energy going in at the beginning, that's the thing that's preventing this whole reaction from moving forward. Okay, we don't have a ready source of energy to break the original covalent bonds, and therefore this is not something that's going to happen to you spontaneously. Right? The energy in here, okay, is what you can call, well, it's what has to be acquired. We have to acquire the energy. We have to reach the point where we can break that bond before we could form the new bonds that are going to give us a greater yield of energy, right? And so here we get to the second concept here. This is what we're going to be reading about, or what you've already read about in the chapters. Chapter 5.13, it talks about this concept of activation energy. And it says that enzymes reduce the activation energy, and thereby they make these reactions happen more readily, right? So at this point, you know that covalent bonds are really things that add stability, and therefore it takes a lot of energy to break those covalent bonds. If we made it a lot easier for these bonds to break, okay? In other words, if we were to create some structural, maybe if we took this disaccharide and held in place and twisted it just slightly so that water molecules could come in and access that bond more easily, it'll be easier for us to achieve the point where the reaction could move forward, right? In other words, if we could reduce this, we could take this energy in and make it a lot smaller, right? We could make it like the energy in, instead of being this big mass, if the energy in was a much smaller amount, energy in, dramatically reducing the activation energy. That's going to make it a whole lot easier for the reaction to move forward. Okay? And that's basically another way of putting what you might have already seen by reading the textbook. I'm going to go through the textbook now. And you could read through this and it says, yes, exactly what he said. We're going to be lowering the energy barriers. And so if you scroll down, we have to achieve this state of high activity. In other words, we have to increase the energy state in order to allow the reactants to go to their final state of being products. Okay, and what's happening in the case of the energy is the enzymes holding the reactant in a way that allows that energy, that intermediate state, that transitional form that allows the reactant to convert to the product. It's a lot lower. We don't need to push it quite as hard in order to get it to the product. Therefore, it's a lot easier for us to get to the product. Okay. And then if you think about it, the next thing that could happen would be this enzyme is going to be releasing the products. Okay. And after the products are released, then the enzyme is right back to where it started. Right. And so this is the standard way of depicting the way that enzymes work. Okay. We're dealing with an original situation where there's a huge activation energy without enzyme. Okay. Whereas activation energy with the enzyme is much lower, and it's much easier for the reactants to achieve that, right? It might take 100 years for all the sucrose molecules to spontaneously reach this amount of energy in order to allow them to become glucose and fructose, okay? But because we're reducing the activation energy substantially, it's happening in a second, right? And so the difference is between 100 years and 1 second. That's kind of like the way in which the reality of enzymes is that they're taking a reaction that would be happening in a forward direction, but taking a really long time to happen and making it happen in a very short amount of time. And in effect, what this tells us is that it looks like the enzyme is doing magic because it's making the reaction happen when it doesn't happen at all in the absence of the enzyme. But the real truth is that it does happen in the absence of the enzyme, but just way too slowly for us to detect any forward progress. Whereas with the enzyme, we're getting a lot of forward progress relatively easily, quickly. Okay. So the next, let me clear my pictures here. The next section in the textbook is the 5.14. Let's go there. Okay. And it's basically telling us what we were going through a little bit earlier in our discussion, which is that the enzyme does its trick. Okay, come on. I have a new sketch pad because my old sketch pad was really kind of temperamental. Okay, and so the what's going on here is that the enzyme, okay, this is actually the same enzyme that we were talking about before, right? The enzyme sucrase is going to be, there's this really specific active site. I mean, this is it's a protein, and this is a specific structure on that protein that is able to bind to the substrate. The substrate is going to bind into that site. And once it binds, we're going to be creating this thing called an induced fit. Okay? Once the substrate binds, the enzyme is going to change its shape a little bit to cause this bond to become more accessible to the water molecules, which are then going to break that bond altogether, right? And so the water molecules doing that, and here you can see the two monosaccharides have been separated from each other, and at this point, all we have to do is to release the products, release the glucose and the fructose, and then we're right back to where we started. Okay? This is a diagrammatic picture of an enzyme that I'm hoping you can relate to because you're relatively familiar with the kinds of molecules that are going that are activated by this. This is a real enzyme that you have in your digestive system because you are able to eat table sugar and you break the table sugar into glucose and fructose. Okay. So let me go back to my other share, which is the whiteboard. Okay. And let me reduce the size here again. Okay, why is it not letting me reduce the size? Okay, well, forget reducing the size. I'm just going to go ahead and continue on here. Okay, we've covered pretty much everything in the top half. Okay, so right down here to the point where it says, how do enzymes have the effect of lowering activation energy? Okay. Now we're going to be looking at some of the other things that are there. For example, we're going to be encountering, as we talk about metabolic biochemistry, the fact that a lot of enzyme catalyzed reactions require consumables, right? In other words, we have to, let's say there might be something like an enzyme. Enzyme might not be able to do the reaction on its own. It might require an enzyme and a cofactor. Okay, and after the catalysis takes place, we're going to end up have the products, the reactants, are going to be converted into products. That part is okay, but the cofactor is going to be altered, right? The cofactor is kind of like one of the reactants. It's functionally kind of reactant. We're going to have an altered cofactor at the end. Well, I'll give you an example of this when we get to that point in our discussion. So a cofactor could be a coenzyme. Okay, and oftentimes these cofactors are touching our lives because a lot of cofactors, things like vitamins, right? Things that are consumables. If you are not taking vitamin supplements, that means all these vitamins are coming in through your diet, and yeah, part of what we get from our daily diets, including vitamin supplements, are the coenzymes required in order to run the enzyme catalyzed machinery that we have in our bodies. Okay. Relating to the lab experiment, and I know that you've got a lab experiment that you've either done or is in the process, you're doing it right now. You've probably done it by now because I've looking at the schedule. This lecture is coming pretty late in the semester compared to the enzymes laboratory activity. And so at this point, you'll know, for example, that enzyme catalysis is sensitive to temperature. It's sensitive to pH. It's sensitive to salinity because you will have done a lab experiment to demonstrate the effect of temperature, pH, and salinity on the activity of the salivary amylase enzyme that we'll be using in our laboratory experiment, right? And so maybe you could answer this for me already. Okay, why is enzyme catalysis sensitive to temperature? Any ideas? Why, yes, that's absolutely right. Okay, temperature is going to have an effect on two things. It's going to have an effect on speed of particles. Okay, and so as long as we're at a temperature where the protein is not affected through some process of denaturation. In other words, if we're below the crucial deadly temperature for the enzyme, we would actually expect enzyme catalysis to increase in speed as temperature increases. And the reason for that is that the enzymes are encountering their substrates at a rate that's proportional to how fast the particles are moving. And temperature is simply a measurement of how fast the particles are moving in that system, right? The difference between a warm glass of water and a cold glass of water is that with a warm glass of water, the water molecules are bumping into each other and have much greater velocity. The particles are moving much faster than they are in the cold glass of water. The cold glass of water, they're just kind of jiggling around kind of slowly, whereas in the warm glass of water, they're zooming back and forth like that. Not terribly useful, so I'll undo that. Okay. Now, when the temperature gets too great, you know that, and here's the other thing that's important to us. Remember that enzymes are proteins. Oops, that's not right. There's no two E's in protein, so are proteins, and therefore they're going to be subject to alterations. If you were to increase the temperature too much, you might actually cause the protein to denature. Similarly, if you were to alter the pH too much, you could cause the protein to denature. And salinity is going to affect the polarity of the water system. And so enzymes that are held in place by hydrophobic interactions are going to be potentially altered dramatically by salinity, right? And so, yeah, I guess if you haven't figured this out yet, the whole enzyme laboratory activity that you guys did is it's really as much about proteins as it is about enzyme catalysis. Okay, in fact, it's all about proteins, and you're only learning about the enzyme catalysis aspect of it right now. Okay, so yeah, there are some other kinds of molecules that are catalysts. There are also RNA catalysts. That are technically not enzymes. They're enzyme-like, but instead of being proteins, they're made of RNA. And we would call these guys ribozymes. And I'm putting this in as a little bit of an aside because it's not technically true that the only catalysts that we have in our cells are protein enzymes. We also have some catalysts that are RNA, and they have some pretty substantial responsibilities in a lot of things, including things like protein synthesis. Okay, now let's go back to the textbook and move forward. And I'm going to gloss through this a little bit, but enzyme inhibition actually is a pretty substantially important topic. I'm going to use my other things, right? And so the two ways that we're going to be finding enzyme inhibition taking place, right, would be one where the inhibitor, if you have an enzyme inhibitor, it's going to be this extra molecule, right? So this would be an inhibitor here, this would be an inhibitor here, and they could differ in two substantial ways. So there are two ways to which enzyme inhibition takes place. Okay, one of which would be simply for the enzyme inhibitor, this molecule here, to have its effect on the enzyme by sitting where the substrate normally sits. In other words, it alters the active site, which would prevent the enzyme from attaching to its substrate. Okay. Now, alternatively, there are other kinds of inhibitors that don't attach to the enzyme, the substrate binding site, right? But they actually cause the enzymes to change their shape by attaching somewhere else. The non-competitive inhibitor would simply alter the structure of the enzyme to the point where it's no longer able to bind to the reactants, right? Okay, now, this is a point that we won't be needing to address this semester, the distinction between competitive and non-competitive inhibition. But you certainly will be needing to return to this concept later when you take classes like physiology, right? Right now, what I want to say is, well, irrespective of the mechanism of inhibition, enzyme inhibition actually is a pretty important concept because for if for no other reason, you might be interested to understand, at least in a very general way, how pharmaceuticals work. Right? I think it's actually in the next part of the chapter. Right? So yeah, this is another interesting point. But what we need to do is to go into the last reading point for this section, and we don't really have any good pictures, but go ahead and read through this. And it just is telling us here that one way in which drugs can work is by causing enzymes to not work. Which kind of makes sense, right? Yeah, if enzymes are these super important molecular machines that our cells rely on, if you were to prevent an enzyme from working as fast as it normally does, that's going to have a pretty substantial effect on your physiology, on your cellular physiology, right? And so I want to give you a couple of examples I'm hoping will be potentially familiar to some of you. Maybe not, but it's worth a try. Give me an E for effort at least. I'm going to erase some of this and do right. I'm not going to erase this. I'm just going to go to a different whiteboard. Okay, and we'll talk about those two that I had described there. One being an ACE inhibitor, ACE inhibitor. Which basically means it's an angiotensin converting enzyme. Okay. Now, what this does, I mean, what this enzyme does, I know it's a mouthful. That's why we say ACE, right? What ACE does is it converts angiotensin into another form, which causes a pretty rapid increase in blood pressure. Okay. And we have lots of angiotensin ready to be converted into another form that's going to cause your blood pressure to increase. And there's going to be some conversion of angiotensin happening at all times, right? In other words, ACE is going to be converting angiotensin into the thing that causes your blood pressure to increase all the time. Okay? Think about that for just one more second, and you'll realize why ACE inhibition would be really good for some population, for one population in particular, of medical patients. Okay? If you inhibit the effect of ACE, you'd have the effect of good for ... Well, yes, as a matter of fact, if ACE's activity has the effect of increasing blood pressure, we've got a huge proportion, a huge number of Americans who are suffering from a condition called high blood pressure or hypertension. And so if ACE normally increases blood pressure, general inhibition of ACE is going to be good potentially for as a treatment for hypertension. Okay, you know, it's far more nuanced than this. You simply can't prescribe an ACE inhibitor for anybody who's got high blood pressure. There are a lot of considerations to make, but the general concept here is pretty easy, right? I mean, how does a drug work? How does blood pressure medication work? This particular blood pressure medication is something you could understand because we know that the normal enzyme that's inhibited by this drug is going to be causing blood pressure generally to increase. All right, that was one example. Now the other example is kind of related to what we've been talking about recently because, well, in the case of a protease inhibitor, this must be some kind of molecule that inhibits protease. And a protease is an enzyme that breaks down protein. Okay. So in other words, it'll take a polypeptide and break it down into smaller chains. Okay? And so a protease would be a digestive enzyme. In a lot of cases, when the food that you eat gets down to your stomach, you've got a lot of proteases breaking them down. That's not the protease inhibitor that I'm thinking about. When you hear the term protease inhibitor, you're probably talking about somebody who's being treated for HIV, an HIV treatment. Is typically modern HIV treatment is typically going to involve some cocktail of protease inhibitors as well as potentially some other antiviral drugs. Right? Now, the reason for this is that in order for HIV to complete its cycle, okay, HIV requires protein cut to be made in its genome in order for it to complete its cycle. In other words, if you don't have the ability to break the protein where it needs to be broken, the HIV will not be able to complete its cycle and it'll simply be short-circuited and maybe the HIV will die, right? So again, the idea here is pretty straightforward. It's very complex in a lot of ways. In other words, the actual practice of creating a drug and administering a drug in such a way that will make for an effective HIV treatment is something that requires a lot of training. But the basic concept of how the protease inhibitor works is actually pretty straightforward. Okay? HIV, the AIDS virus, requires a protease in order to complete its cycle. If you don't have that protease, the HIV dies. And so the protease inhibitor is going to be a great drug to use to treat HIV treatment, provided that it doesn't have any other effects on the patient themselves, right? So that's kind of it. I think I've covered everything that I need to cover with this video. You've got a good idea of what enzymes are. They're proteins, they're catalysts. They work by lowering activation energy. They work by attaching to these substrates that they need to carry out catalysis on. And the inhibition of said enzymes is one of the important ways in which drugs carry out their effective, the beneficial effect on patients. Okay.